Once we know the basics of the biochemistry of photosynthesis, we can start to take a look at the environmental controls over differences in photosynthetic rates. We're going to start with light response curves. So before we had talked about gross photosynthesis, and now we're going to talk about net photosynthesis. Now, net photosynthesis is the net rate of carbon gain measured at the level of individual cells or leaves. So when we take a leaf and we measure the photosynthetic rate of that leaf, there are different processes that are happening. Part of what's happening in a leaf, at least in the light, are photosynthetic reactions, and that's the gross uptake of carbon. But at the same time, there's respiration that's happening within a leaf. And it's really hard to try and separate how much respiration is happening in a leaf while it's also photosynthesizing. So from an empirical standpoint, we tend to just take a look at net photosynthetic rates, which is the net rate of carbon gain measured for individual leaves. So just on a net basis, how much carbon is it taking up, for example? Part of what we also want to understand when we're thinking about net photosynthesis is the light compensation point. So what is the minimum light level required for photosynthesis to occur? So often, plants are in relatively shady spots. So plants that are in full sun and brightly lit might not be limited by light. But here, in the understory, you'll find different plants that tend to do a little bit better at lower light levels than other plants do. But what is the lowest level at which they can actually photosynthesize? How much light do you need to have a net gain of carbon? To figure out questions like that, we can use light response curves. Typically what is done is that photosynthesis is measured on a leaf, and we'll talk about how that's done later. But we measure the rate of photosynthesis, and what we do is vary the light level, tend to start at relatively dark and bring it up to brighter and brighter light levels, and then bring it back down. So on the x-axis, we're going to have irradiance. On the y-axis, we're going to have net photosynthesis. Here the units tend to be micromoles per meter squared per second, but it's the amount of carbon that's fixed per meter squared of leaf per second. Now that doesn't mean that we actually have a square meter of leaf and we don't measure it every second, but that's just a standard set of units that we use. And we had talked about whether you want to use energy content or biomass or carbon, and here we're going to use micromoles of carbon or micromoles of carbon dioxide. A typical light response curve looks something like this. You have a given irradiance where net photosynthesis is 0. Any darker than that, you have net release of carbon from the leaf. Any brighter than that, you have net uptake. Now, as it gets brighter and brighter and brighter beyond the light compensation point, you tend to have a relatively linear increase in net photosynthesis. And over this range, we tend to think of photosynthesis at this scale as being light limited. So we initially have a linear increase. Then it starts to level off until you get to a point where additional amounts of light don't actually generate more photosynthesis. But nor does it lower photosynthetic rates. And this is a point where you're at light saturation. And we talk about that point as being, we want to know how much light it is. And in this case, it's 1,000 micromoles per meter squared per second is the unit that we'll use for radiance. And somewhere at about 1,000, you have light saturation. This point right here is your maximum rate of photosynthesis. Now, once you've hit light saturation, additional amounts of light do not increase photosynthetic rates, and after a certain point, photosynthetic rates start to go down. That's when you start to have what here is termed photooxidation, but is the same as photorespiration for all intents and purposes. So we have two portions of the light curve. The first is that the portion of the curve where you have light limitation, and that's when you see a linear increase in photosynthesis with changes in irradiance. The other part is where photosynthetic rate is really limited by how much CO2 you have in a leaf. So adding additional light might give you the opportunity to fix more carbon, but you just don't have enough carbon in the leaf, is generally considered. And photosynthesis is limited by the rate of carbon fixation, which tends to be limited by the concentration of CO2 within the leaf. Now plants adjust the components of photosynthesis so the energy trapped by light harvesting reaction closely matches the energy needed for the CO2 fixation reactions. What that means is that it's not necessarily beneficial to be light limited, nor is it beneficial to be limited by carbon fixation. If you're light limited, you probably haven't invested enough in light acquisition, and you might have invested too much in carbon fixation. And so plants shoot to be somewhere in the middle, not necessarily strictly limited by light nor limited by carbon fixation. And you can reallocate resources away from carbon fixation to light acquisition if you're in the light saturated part too often, and vice versa. So plants tend to try and adjust the components of photosynthesis so they're at somewhere close to the inflection point. Now where that is and where a light compensation point is depends on the species. So if we say roughly that full sun is about 2,000 micromoles per meter squared per second, it's somewhere in the intermediate light levels that a plant will typically operate. And often it has to be down here also. And we'll talk a little bit later about how low a light level photosynthesis can actually occur.